and they are true academician and researcher, and they are truly a stalwart. So it's an honor to introduce Dr. Professor Anju Grewal. Ma'am is a professor at Dayanand Medical College and Hospital. She has a vast experience of looking up the journal as editor-in-chief of Journal of Anesthesiology and Clinical Pharmacology. She had just given a handover, but she has done a wonderful work for GOCP, taking to a great height. We have Dr. Jayashree Sood, ma'am, who is Indian College of Anesthesiologists CEO. She is head of the department at Sir Gangaram Hospital. She is a dynamic lady, mentor, and teacher for many of us. We have learned, we have grown up hearing her at many platforms. Welcome, ma'am. We also have Dr. J.V. Divetia, who is Professor and Head, Department of Anesthesia, Critical Care and Pain at Tata Memorial Hospital, Mumbai, India. He has been in a capacity of at various platform of repute. He has been the past editor-in-chief of Indian Journal of Anesthesia, and he holds reputed posts of president of ISCCM, IDA, and so on and so forth. Welcome, sir. The next chairperson is Dr. Kirtina Saxena, again, one of my teacher who has trained me during my MD time, and still I'm learning from her. She is director, professor, and head at Maulana Azad Medical College, and she is one of the wonderful teacher. Welcome, ma'am. We also welcome Dr. Syed Mohit Ahmed, who is professor and ex-chairman of Department of Anesthesiology at Aligarh Muslim University. He's a dynamic person. He has been the chief editor for Indian Journal of Anesthesia, Central John, and he has been involved into many academic, clinical, and research activity. Welcome, Dr. Syed, please. So I hand over this platform to our eminent chairperson for the conduct of the session, please. Over to you all.
Thank you, Dr. Rakesh, for such a generous introduction of all of us. It is indeed a pleasure to introduce you as the first speaker of today's academic proceedings. Dr. Rakesh Garg is, as has been, you all have heard him, he's an additional professor of the Department of Onco-Anesthesiology and Palliative Medicine at the All India Institute of Medical Sciences, New Delhi. He has been a long associate with me, and as I hand over, I will always have very fond memories of you assisting me so ably as an associate editor of the Journal of Anesthesiology and Clinical Pharmacology. He is also the associate editor of the Indian Journal of Anesthesia, with areas of interest at onco-anesthesia, airway, research, pain, and palliative medicine. He has over 260 publications in both national and international journals, and has authored more than 25 chapters, and hence he is the correct person to be talking to us on type of studies and selection of a particular study design. Over to you, Dr. Rakesh, for the talk.
Thank you so much, ma'am, for your kind words. You have always been a mentor and guide to us for various publication-related issues, and you have been truly mentoring many of us for learning purpose. Thank you so much. So I think the first session we are taking today is the type of studies and selection of a study design, which is one of the very important aspects when we start thinking of that for answering a particular research question or a clinical problem. We have arena of study design and which should be choosing for that particular, answering that particular research question. I bring greetings from my institute. So when we say the study design, probably we are looking for publications and we are looking for various purpose, but the basic theme for this session and the session by the other speakers also would be that we are contributing something to the society so that they can use those academic things in a more comfortable way. Now, when we talk of the research process, we know the cycle of the research process. We review the literature, we formulate a research question, we select an appropriate research design, we go and collect the relevant data, interpret the findings, and then finally publish their findings also. And then again, it becomes a review of literature for somebody else. So among this, one of the important aspects is whenever we have a research question, which study design would be the most appropriate for answering that research question which we have formulated based on our review of literature. So when we say study design, it is a specific plan or a protocol for conducting the study, which allows the investigator to translate the conceptual hypothesis which we have developed based on the research question, into an operational one, so that we can get some data and then we can interpret and bring out some conclusion from it. So when you talk of study design, it primarily we say that if you understand the study design, before I proceed further, if you talk about the research particularly, we divide the research as primary and secondary research. And the primary research could be basic clinical trials, epidemiological trials, and secondary research is the conglomeration of the already published data into some form of narrative or a systematic review or a meta-analysis in addition. So this is what the research is there. And the primary research we are talking about, which will be inclusive of various study designs, they could be depending upon whether it's basic clinical trials or epidemiological. But primarily we are talking of in our clinical practice of anesthesiology, critical care, pain, medicine. We are usually talking of various experimental and observational studies. But yes, some of the researchers are also talking about the basic research because it's a very important one. So when you talk of the observational and experimental studies, primarily in a very broad overview, the observational studies could be descriptive or analytical, and experimental studies, which we usually perform, will be of randomized controlled trial, which usually has a higher level of evidence. So when we talk of any study design, whether for the purpose of understanding and bringing into our clinical practice, we should be very, very clear that there is some level of evidence for the various studies that are being published. So whether they are single case reports or we are going up the ladder as the point of level of evidence, going to the case control or case cohort, as we go up the ladder, the level of evidence increases. And the reason is basically more robust the study design is, more acceptable it would be for taking into a clinical practice. And that's why we say the level of evidence increases as we go up into this ladder of hierarchy. And obviously, we use the term the filtered data or filtered outcome versus unfiltered. So till the level of randomized controlled trial, it is basically based on a particular subset of population. So it remains a little unfiltered one, it becomes focused one. But if you go for systematic reviews and meta-analysis, it becomes more filtered because we are analyzing each RCT from its quality point of view. And hence we are filtering that what were the lacuna or the missing points that probably will downgrade the level of RCTs also. That's why meta-analysis remains the highest level of evidence, because we are not only analyzing each individual study, but also we just combine the data or pool the data to have a meaningful outcome for the purpose of clinical practice. So let's see some terminology which are used in the study designs. So the most commonest word that we use in the study design is whether they are prospective studies or retrospective studies. So when we say that the study or the data collection is happening at one point of time, whatever it is the cross-sectional study. And when we go backward to collect some data from any resources, it becomes a retrospective study. And when we proceed forward, it becomes prospective study. Again, in some specific type of situations, we can go forward and backward, and it becomes the ambidirectional study. So these are the four terminologies that we will be using in the study designs to confirm that study design conforms to these aspects of data collection. For example, if we see this study where the authors have collected the massive blood transfusions and its association with the post-operative complications in orthopedic surgery. So what they have done is they have collected data from the cases which has already been done from various records, files, or electronic system and try to analyze it. So it becomes a retrospective study because we are not proceeding further. We are just collecting the data for the subjects or the participants who has already undergone that particular procedure. So it becomes a retrospective study. But if we try to follow certain outcome or certain observations in a further time period, it becomes a prospective study. For example, in this, the authors want to look for the critical incident in pediatric anesthesia practice over the coming time, and they're collecting all the data prospectively. So it becomes a prospective study if we are going forward. At times, we do ambidirectional studies. For example, if some technique or some procedure or some drug is being used, but then some new data comes and then the gold standard or standard of care changes, but you want to compare them. So you cannot be using the outdated study technique or the drugs which has been found inferior, because you know this is better from some of the studies, then you try to compare some data from the patient who has been used with the older drugs or the older techniques and try to compare it. For example, say we have been using the video assisted surgeries and the robotic assisted surgeries. So if the robotic assisted surgeries become the standard of care, then those situations, you may not be randomizing the patients into video assisted surgeries, and then you may pick up the patients who has undergone VATS in the recent past, and then subsequently you can do robotic assisted for the prospective one, and it becomes an ambidextrous study. The other study design that we commonly use is the cross-sectional design, where primarily you are going to answer that actually what is happening. So in this type of study design, what you are doing is you start at a particular time, you select a study population, and at that time itself, you are not going in a backward direction or a forward direction. You just select a population, and from those population, you try to find out which patients have the disease and which patients do not have the disease, and then try to analyze the various parameters or the etiological factors or anything else, that some factors were present or absent in patients having disease or outcome, or versus no disease or the outcome with no outcome. So that is what you are doing it at one point of time. So it becomes a cross-sectional study for that particular study design aspect. The advantages are many. It is quick, is inexpensive. They can study several exposures and outcomes at one point. It provides prevalence, and it helps to generate the hypothesis. This is one of the important aspects, specifically when we do not have some robust data. But it cannot infer causality. It is prone to bias. No good for rarer disease, and it's not good for disease with short duration and high fatality, because we'll be missing those patients from the population which have selected. Like, for example, this study in which they want to study about the patient satisfaction in emergency procedures undergoing surgical aspect. So what they did is they just selected the patient population and then try to look for the patient satisfaction who has undergone surgical procedures. Now, the next study design at times we use is the case control design. It is to answer for a research question when we are answering what happened. So here what we are going is, we are going a little backwards. So what we do is we choose a study population of our interest based on the research question, and then start to look for in this study population that what was the patients with some disease or some not having no disease. And then you go back and explore that what factors were present or absent for this disease to happen or not disease to happen in a backward direction. So this type of study designs are case control design. Sometimes we try to confuse it with a study design where we have one group as case and one group as control, right? So, or maybe a study and control. So those study designs are RCTs or something like this. So those are not case control designs. So we should not be confusing with those. Herein, we are selecting a study population with disease and without disease and try to find out the factors attributable to disease versus no disease, and then make some meaningful outcome of it. For example, if you see the case control study example, if you want to look for the association between alcohol consumption and breast cancer, so there will be cases which will be females receiving treatment for breast cancer in a particular hospital. And we want to control, we can look for females who are attending the other part of the hospital so that we can compare them, whether there is some association between breast cancer versus alcohol consumption while we compare case versus controls in this. Now, issues in this is when we try to collect data from the hospital controls, there would be some restrictions of the eligibility criteria or the exclusion criteria, and will not be able to extrapolate this to the whole general population. Sometimes we use the external validity of the outcome of RCT. So this means we are not able to generalize the outcome when we are picking up a very specific population who are coming to the hospital to make it generalized to the other population across. The advantages are many for case control studies. It is easy and cheap. We can do it for rare disease as compared to the previous cross-sectional studies. It can study multiple exposures for single outcome. It don't have losses to follow up, but it is restricted, it's limited because it has certain ingrained bias into it, which cannot be avoided. Appropriate selection of controls becomes very, very challenging because you have to match certain factors which can affect the outcome. It is not suitable for rare exposures and cannot measure prevalence or incidence of disease. The third type of study design that we use in our clinical practice is the cohort study design. in which we are going to answer of a research question which is related to what will happen. So in this, we do is we select a study population which is free of disease, and then try to divide them with subjects who are having certain factors responsible for disease and factors which are controls, which are factors are absent related to that particular disease, and try to follow them up with occurrence of disease versus no disease. So this becomes a cohort study design where a population of a cohort is selected and they are being followed up for various factors which attributes to a particular disease. Now, this cohort study can be a prospective cohort study, or it could be a retrospective cohort study also. So in a prospective cohort study, we measure the exposure and the confounder variables at a baseline, and that the patient is then followed up. Some of them will get exposed, some of them will not get exposed to the certain parameters which you are looking for the outcome. And then you follow them up and then look for that how many of these patients have the outcome versus no outcome. On the other hand, retrospective study, we look back to the time event and starts looking for the events that have already occurred. So in this situation, we go back, though this will be the usual procedure, but here the study begins at the level of outcome itself. And then we try to follow them up in the sense of exploring them for the patient who were exposed to certain parameters versus who were not exposed, and then take it backwards and try to analyze the situation for a particular outcome parameter. The advantage of cohort study, we can look for the temporal sequence between exposure and disease. It's well suited for assessing the effects of rare exposures. Incidence of disease can be calculated. It can examine multiple outcomes. It can do estimate of risk, and it shows the natural history. But the disadvantage, it requires a lot of time and money, and subject attrition or loss to follow-up can happen. And certain bias can ingrain into this, so it becomes an issues, and we should be considering these bias while we are collecting and analyzing the data accordingly. Now, coming to the interventional studies where some intervention is done or not. If there is no intervention done, this means this is an observational studies where, again, based on the comparison group, it could be a descriptive study or analytical study. On analytical study, based on the direction of study from exposure and outcome, if exposure and outcome are being done at the same time, it's cross-sectional study. If we are moving from exposure to outcome, cohort study, I just mentioned. And we are, if you're following back from outcome to exposure, it becomes a case control study. Experimental study, some intervention is being done. If the group is being randomized versus non-randomized, it becomes non-randomized control study or randomized control study. Now let's see the study where breaking the midnight fast. This was an observational cross-sectional audit of a preoperative fasting policies and practice. So here the authors have collected the practice patterns in a cross-sectional way of all the patients about the fasting procedure in patients who are undergoing surgery. So it becomes a cross-sectional study, observational study. In case if you want to compare, for example, they want to compare propofol and midazolam for arousal time from sedation after spinal anesthesia for any infraumbilical surgeries, it becomes interventional study because they are intervening by use of propofol and midazolam. If they are using dexmedetomidine infusion on propofol requirement, again, it becomes because they are using the drugs as an intervention versus some control. So it becomes an experimental study. Now, experimental studies, they are the gold standard for determining the relationship between factors and event. And these are the prospectively done with a control group so that you can have some meaningful outcome. So when we say the experimental study design, this is how we follow in the experimental study design. We plan a study, we choose our study population, we randomize them into intervention and control. Now, randomization has two aspects here. One is generation of randomization sequence, and second is allocation concealment. So it has both the components. It should be followed for this type of studies. And then both the groups are looked for outcome of interest versus no outcome being followed up. And then we try to analyze those outcomes. The steps that usually follow for these studies is we draw up a protocol, a reference population, a sample population. We do exclusion, inclusion criteria for these patients, and then randomize them into two groups. We give them certain interventions and then follow up and then make assessment of the outcome. So this is the basic overview of steps. Now, in these clinical trials, there are two types of designs that we are usually using, parallel group and crossover. What we do in parallel group is participants are screened for the entry criteria based on inclusion and exclusion parameters. And then you randomize them into control group and the experimental group, and they are there followed up for the outcomes, and then you analyze the data. So this is a parallel group design of an experimental study. In a crossover design where you think that the patient population may be influenced by their demographic profile and all those things, you try to do a crossover design, wherein the participants are screened for entry criteria based on your inclusion and exclusion criteria. You do a randomization and allocation concealment into control group and treatment group and look for the outcome parameters. Now, after some washout time, this is a washout time that will depend upon what type of intervention has been done, so that the patient is free of the effect of the previous intervention. So that is what the washout time period should be followed. And then the patient who received the control now becomes the experimental group, and patient who was in experimental becomes a control group. And then again, they are followed up. So this is a useful study where you think that individual patient will be affected by various baseline parameters. Then you try to have a crossover design and try to follow them up. Now, the RCTs can be of various types. It could be before and after comparison, comparison with placebo and standard of care, two medicines, procedures, tests, or techniques. So this is how, or sometimes you can have more than two groups also. The advantages are many, because when we do a randomization and allocation concealment and we take care of blinding and all those things, which we'll discuss in subsequent sessions, the chances of various unknown confounders gets eliminated. It provides a strong evidence of cause and effect, allows standardization of criteria, and we can use strong statistical methods which are built on some assumptions which are being followed by RCTs. They're expensive. That's a limitation. They have, we cannot have many research questions that can be answered by one RCT. And there are certain situations in which we are not very pragmatic. We become a little artificial because we have certain inclusion-exclusion criteria. Now, control groups are important because certain diseases are self-limiting. They fluctuate, the disease severity, placebo effect, Hawthorne effect, regression to mean. So these are the things where we need the inclusion of a control group. Now, when we say the uncontrolled study, the disease will have a treatment effect where the total treatment effect may be the true one, or it could be because of other reasons like regression to mean, self-limiting, or placebo and Hawthorne effect. But when we have a control group, the outcome, the treatment effect will be there in both the groups. So that will be somewhere balanced, and that's why this will be eliminated in both the groups. And when we do a good randomization for these patients, the confounders are also taken care of. And follow-up and compliance in the study design will be same in both the randomized groups. So the difference between the treatment effect S and treatment effect Y will be the true treatment effect, because the factors which can affect the outcome are being balanced in both the groups, because we are doing randomization, we are doing other basic nitty-gritties of doing a study, which includes blinding and all other aspects. That's why the RCT remains a gold standard studies, because it provides an unbiased and precise estimates obtained from various RCTs. But now question is, having said that, how to choose a study design from these many study designs we have. So it depends upon ethics, logistics, economics, and scientific thoroughness to understand that what research question are we going to answer. So appropriate study designs will ensure the validity of results, and that is how we need to be very, very sure that the potential bias, the confounders, and vulnerability are taken care of by that particular study design, and hence we can choose them. Now, depending upon the research question, this will help us to understand that if we are doing a novel study where we don't have any data, we don't form a hypothesis, basically the case report series and descriptive epidemiology helps to form the hypothesis for you for subsequent studies. And once certain hypotheses are made by certain data from it, or maybe animal study or lab study, then we do a clinical trials, and that is for the hypothesis testing, so that you can have some meaningful conclusions. So this is how, depending upon review of literature, which I mentioned in the beginning, will help you to understand how the things will go on. And finally, this is the crux which will help you for making a good or selecting a good study design, is based on aims, objectives, the research question, and hypothesis that you want to prove. So in a very brief overview, if I can summarize, if you want to answer the burden of illness with prevalence and incidence, probably the cross-sectional studies or longitudinal survey will be good. If you are looking for the causation, risk and prognosis, case control studies, cohort studies, or RCT will be good. If you are looking for treatment efficacy or diagnostic test evaluation, it will be an RCT that will answer your research question. If you want to look for cost effectiveness of a study, again, RCT will be a good option. So to summarize, looking for the various studies, depending upon what you are looking for, based on your aims, objectives, and research question, the descriptive studies helps to make you develop an hypothesis. Case control studies investigates the relationship to outcomes. Cohort study define the meaning with the exposures, and clinical trials test the link experimentally, and you can make some meaningful conclusion. So this was in brief about the various study designs and how to choose them depending upon what review of literature we have done, what was the aim objective, and what research question we have developed. This will help us to choose a study design to answer the research question well. Thank you so much for patient listening.
Thank you, Dr. Rakesh, for the wonderful elucidation of an important talk in such a short time, but you have made it quite interesting and simple. I hand over the mic to Dr. J.V. Devatia, sir, to introduce the next speaker.
Good evening. Thank you very much for that lecture, Dr. Rakesh, because that leads us on to the next topic. Is once you've decided on a study design and how you want to do a study and what you want to study, there are some very important regulatory aspects that you need to follow, some permissions you need to take. And to speak on that aspect, we've got Dr. Colonel Rakhi Goel with us. And she's the lead consultant in anesthesia at the Madhukar Rainbow Children's Hospital in New Delhi. She was formerly the professor at the Army Hospital R&R in Delhi and AFMC Pune. She's an associate editor of the Journal of Anesthesiology Clinical Pharmacology. And so I invite Dr. Colonel Goel to speak on the given topic.
Thank you, sir. Thank you so much for the kind introduction. I have my talk recorded and I would like to play that. Good evening, everyone. I am going to talk about approvals and registrations that you require for conducting a study in anesthesia. So the frequently asked question is, I want to start a study. That's what most of my PGU students used to ask me. Where should I start? How should I start? Shall I just make a protocol and start collecting data? Do I need any permission? What do I do? So first you plan well to make the study successful, plan well, execute well, one step at a time, and no shortcuts. By shortcuts, I mean your approvals and certifications should be correct and at the correct time. So let's come straight to the approvals. The first thing that you need would be the approval of the ethics committee. Then would be the registration in the Clinical Trial Registry of India. I'm not talking about the rest of the registries because here I assume that we're talking about all the trials, all the studies that we do in the field of anesthesia, including ICU. So now, what is ethics committee? It can be your institutional ethics committee, which is there in your hospital, in your medical college, in your institution. It's also called as institutional review board. Or if you're not part of a certain institute, you could also be part of an independent ethics committee. There are several independent ethics committees which are just similar to your institutional ethics committee, and you could enroll there. Now, the most important thing here is that the ethics committee should be registered. Now, you can't just have a group of people calling themselves the ethics committee of the hospital and giving approvals to students. No, it has to be registered with the Drug Controller General of India, the DCGI. And I mentioned here the website where the hospital or the institute people could go and register themselves. Now, accreditation of this ethics committee is mandatory since 2018. Without that, the ethics committee is not valid. Now, this ethics committee is basically because the human subjects need to be protected. The subjects on which you are trying to do any study, you are trying to see outcomes to whatever study you have planned, where the highest level of ethical standards can be maintained. Now, this follows the general clinical guidelines given in the ICMR, the good clinical practices. This is all mentioned in the ICMR booklet. Now, this committee has a particular composition. The most important is the chairperson, who has the final say. Now, this chairperson is not from the institute where the ethics committee belongs to. Chairperson is an outsider. The member secretary, I mentioned at the end, but he's the most important guy. He is from the institute and he kind of coordinates the whole ethics committee. Rest of the members, 1 or 2 from the basic medical science, 1 or 2 clinicians, 1 legal expert. Now that's very important. You have to have a legal expert to add legality to your authenticity to your ethics committee. One's NGO representative or a social scientist, 1 philosopher, and 1 complete layperson from the community. So that in very basic terms, you understand what, or they understand what you're trying to do. For now, all these 12 people need not be physically present when you're proposing a protocol in the ethics committee. At least 5, and I've enumerated these 5 people are mandatory. Now, in the last 1 year, because of the pandemic, the regulation from the ICMR was that a couple of people may be on virtual presence, but some have to be physically present. I'm sure this is going to go back to all physically present, at least 5 during the ethics committee meeting. So typically, these proposals are sent to each member in advance. So you make a protocol, you send it to your ethics committee, the member secretary distributes your protocol well in advance to all the members so that they have time to go through it. And in some institutes, we have a scientific committee meeting sometime before the actual ethics committee meeting, so that the details, the scientific details of your study are discussed well in time before the ethics committee. So the ethical part of the methodology is discussed during the actual ethics meeting, just to save time. Otherwise, you could discuss your protocol in detail in the ethics committee. So these days, you typically make a PowerPoint presentation and you're given a chance to talk about your study, and then people ask you questions which you must answer satisfactorily. Now, there's some clarifications here. If it's a multi-centric trial that you have planned, so 1 ethical clearance is not enough. It has to be from every center. The ethics committee of individual centers have to give the approvals for a multi-centric trial, even if they have the same protocol. Now, at any time during your study, if there's a change in protocol, or if there's a change in the place of the study, or any changes in the principal investigator or the co-investigators, you got to inform the ethics committee in writing. Now, once you start your study after the approval, you have to give 6 monthly reports. Suppose your study is of 2 years, so you give every 6 months your report that how is your study going. And then at the end, give a final report. Now, over and above, the ethics committee members have the right to monitor your trial. So that's part of the deal. And you have to accept. Now, when you submit your proposal to the ethics committee, everything should come over there. The protocol title, your principal investigator details, language, which means commonly it's bilingual. The layperson should understand, and all your subjects, participating subjects should understand. So the consent forms, your protocols, they are supposed to be bilingual, usually English and Hindi or the regional language, wherever you are. the details of the study, what is going to be the role and responsibility of the participants of the study, the risks and benefits of participating in the study. Now, this has to be amply clear, especially in your consent form. And also you got to put in writing the alternative treatment options available so that there is absolute clarity of what you are proposing to do with the participants of your study. Now, all this information is required so that the ethics committee is convinced that you are going to conduct your study in the most ethical way without any intent or chance to harm any participant. Some other things that you need to inform during your protocol submission is the cost, who's sponsoring it, where you're going to get the money or the drugs or the device or resources required. What about the compensation for injury? If there is any adverse effects, who compensates the participants? All this has to be in writing at the time of protocol submission to the ethics committee. Confidentiality, most important. Data protection in today's time, extremely important. You have to commit that you will keep the data confidential. And of course, you have to again put it in writing that the participants will be voluntary. You will not force anybody to participate in your study. Now, a typical consent form would have all the things which I mentioned, and it's printed bilingual. This is just a prototype I picked up from the internet. A sample consent form in English and in Hindi. This is just one page. So this is part 1, part 2. So it's a pretty detailed informed consent form, which you have to produce, which you have to show to the ethics committee. Plus, how will you collect your data? So information, data collection sheet, everything. Protocol in detail, what happens, what is the alternative, everything in detail you got to put up to the ethics committee. Only then they give you first a provisional approval with their comments and any changes or any extra document that they want from you. And finally, as in the right here, they give you a final approval letter. So this final approval letter will have the signature of the member secretary. It will have an ethics clearance number with the date, the contact details, so that if as an outsider I have to check back about your ethics approval, I can contact your member secretary or the chairperson. Now, after you get the approval of your ethics committee, next is you got to register your study in the clinical trial registry. Now, I have taken it from the site so that it is clear that we are here talking about registry of interventional trials, which include but not restricted to drugs, cells, surgical procedures, devices. Devices would include most of our anesthesia devices, process of care changes, preventive care. So it almost includes everything. So whenever we are dealing with any of these, we got to register our trial. Here I am not talking about the drug trials, the phase 1 to phase 4 trials, because in the field of anesthesia, we are actually not doing. That's the pharmaceutical company. So we are not touching that in this talk. Now, thankfully, it is on an international level that the WHO has the clinical trials registry platform. Now, there are these 7 primary registries in the world, and there are 2 partner registries. So the US and the Europe, they form the partner registries, whereas India is part of the primary registries. So we have the Clinical Trial Registry of India, which is affiliated to the International WHO Trial Registry platform, and we are a primary registry. So we can directly register into CTRI, Clinical Trial Registry of India. Now the face of the US trial registry called the clinicaltrials.gov looks like this, while the CTRI looks like this. When you just type CTRI, this is what you get. I'll come to it. Now, why are we trying to register our study? To bring in transparency. Anybody who gets to register is making his or her study data transparent. Study protocol results transparent. Anybody can access it once the study is over. It can, a central body can regulate and streamline the clinical trials. So there is a method that is universal. There's more accountability of what you're doing. Accessibility, people can access your study. All this is with an aim to reestablish public trust in clinical trials. What is the point of doing a study if nobody believes you? So bring in transparency, accountability, centralize the concept on a common international platform, and at the same time, protect the rights of the participants. So coming to CTRI. Here you got to register yourself first. So you get a username and password. So you log in. When you log in, you get a page like this. This is just an example. So it shows you your previous registry trials, your trials under review stage, your completed trial, just terminated or suspended trials. And of course, if you have to start a new trial, you click here to get into a new trial. So when you click there, you get this page. Now you should understand, this registry is not just for studies in anesthesia. So it could be a pathology, PSM, any field, study of any field. So it's very general. And it can also be accessed by people who are not doctors. So a complete layman can access. So there is a public title also. There are a lot of heads which you may not connect to at the first go. But trust me, if you read it in detail and there's this help icon, the I, if you click here, it will tell you how to go about it. The first time, I must confess, I had a lot of problems. And next time, it was much easier because I understood how to go about it. Now, what is the timeline? The moment you submit your registry in the clinical trials, it goes to a set of experts who go through it, and anything which is out of place, they tell you. So you give you a chance to update, edit. So you have to log in and see. You'll probably get a message or an email that you need to update some inputs. So you do that, and after you have finally submitted, within week, 10 days, they process it. If you're not getting their approval, you may contact them through email or their numbers, which are available on the net. Now, CTRI typically wants a lot of data to be put about the study, like place of study, what's the timeline, what is exactly you want to do, what are the drugs involved, what is the review of literature, and things like that. Whole lot of information. But there are 20 items that has those have been marked by the WHO. They are mandatory to be filled. Only then you get a universal trial reference number. Now, this reference number is the magic number which is going to be quoted in the future. So you pick up any good journal, index journal, and see any article, it would be mentioned that this was registered under this registry with this reference number. The moment you put that reference number in the search option, the entire detail of the study comes in. So this is also helpful when you actually submit your manuscript to a journal. The peer reviewers or the journal editors will quickly check back from your registry whether whatever you are putting is correct or not. So this is important. This is just to show you what all information they would want in CTRI. So the asterisk fields are mandatory. Now, the most important question, which study to register? We always think that the interventional the RCTs, they need to be registered, but no, I'll come to it. It's much more than that. Fees? No, it's all free. It's all online. There is no paperwork except for the ethics committee approval, which you are supposed to scan and upload. So everything is online, and timeline, as said, the earlier you submit it completely, the earlier you get the approval. So you get the ethics committee approval, then you get the CTRI done, and only then you start your study. Now, obviously, you're going to have many more questions. So go to the FAQs on the CTRI site, and it's a whole lot of questions which are answered in detail very clearly. So do not hesitate to clear your doubts from the CTRI site. Do not believe your advisors, like your friends, colleagues, who have told you that registration is not required. Believe me, most people think that most trials do not require to be registered, but they do. So please check on the CTRI site before you decide that you do not require a registered trial. Here, this is from the FAQ. I've taken a screenshot. It clearly says interventional and observational. Both studies require to be registered. Now, is it mandatory? If it's a purely observation study, well, it will be required from, say, like you have the US FDA, from the drug licensing authorities, ethics committees, and most important, the journal editors. Any good journal would definitely ask you for their registry number. Well, WHO says, when in doubt, register. Same. I would also say, when in doubt, just register. Just takes about an hour or so to register. And what is there to hide? Nothing. Now, from 1st April 2018, CTRI registered only prospective, no retrospective registration. So please, please, the moment you get the ethics clearance, you first register. Even if you have to collect retrospective data, you have to first register and then start your study. Then recruit the first patient after you get the registration number. Now, this is the most important take home today. This is same thing, what I've been saying. I'm doing a retrospective data collection. And can I be registered? No, you have to first get registered and then initiate data collection, even if the data collection is 5-year-old. So there is absolute clarity on this. So I think you should not waste your time. Just get started, make your study protocol in detail, take help from your mentors, from your seniors, from your colleagues, get it approved by the ethics committee, get it registered with CTRI, and then you are good to go. Thank you for the patient hearing, and I would love to take questions from you.
So thank you, Dr. Raki. We will now move on to the next topic on basic concepts in scientific writing. And I will ask the next chairperson, Dr. Kirtinath Saxena, to introduce Dr. Zulfikar Ali.
Kirti, are you there? Dr. Divetya? Dr. Divetya?
So it is my pleasure to introduce Dr. Zulfikar Ali. He is so Dr. Zulfikar Ali is the additional professor in the division of neuroanesthesia at the SKIMS Sora in Kashmir, and he's in charge of the neurosurgical intensive care unit. He has received the ICA President's Appreciation Award in 2015. He's very well published and very active in publishing and with journals. He was an associate editor of the Indian Journal of Anesthesia. He is an associate editor of the Indian Journal of Anesthesia. He's a co-editor of the Northern Journal of the ISA. He's been a past executive member of the ISNAC, the Neurosurgical Society, and again, several publications and presentations. So may I just request Dr. Zulfikar now to speak on basic concepts in scientific writing. Zulfikar.
Thank you, sir. Thank you, sir, for an excellent introduction. Before starting, I would like to thank the Indian College of Anesthesiologists for organizing this excellent series of webinars, which will enrich the academic activities across the country. I would like to thank Dr. Radhakrishna, Dr. Sanish, and Dr. Rakesh for giving me a chance to be a part of this academic event. My topic today would be to discuss briefly the basic concepts that are involved in the scientific writing. The learning objectives that I will try to focus today would be to identify the elements that are essential in writing a scientific paper. And then I will try to briefly discuss as to what should be the structure and writing style of each of these elements once we start writing a scientific paper. All of us know that once we start the research process, we start the process by formulating a question, and then we select an appropriate research design. We proceed with collection of the relevant data and try to interpret the findings once we compile the results. But the final step, once we finish with our research project that we have taken, it takes its culmination once we try to publish the findings. So a proper scientific communication, once we publish it, it has to have three basic elements. Any good scientific communication, any good scientific paper has to have, it demands precision. We should precisely use all the words that are written in the manuscript, and the whole manuscript should have a clarity and an economy. So if we see the various types of articles ranging from the systematic reviews, randomized controlled trials, and original studies, down to the case reports and narrative reviews. Our focus of attention today, in today's talk, it will be mainly on the original research. And in the original research, what we follow is the IMRAD structure. Most of us know that once we follow the IMRAD structure, we divide the scientific manuscript into introduction, methods, results, and discussion. Meta-analysis follow a different format, which we will try to discuss briefly at the end of the lecture. And similarly, case reports and narrative reviews, they have less structured formats, which I will try to summarize at the end of the talk. Now, if we come to the original scientific paper, the first phase of the paper comes in the form of the title. This is the part of the manuscript that is read the most. And this is the part of the title that is read first. So once we start writing an article, we have to ensure that we have an appropriate title. So devising an appropriate title would mean that we neither have to have a too long title, which may be cumbersome for a reader to go through, nor it should be too short, which does not convey a significant meaning. And once we devise a title, more important is that we have to take those keywords that once an author who is willing, who is going to work in the relevant field, once he searches in, puts appropriate search items in the electronic search media, our publication should be retrieved easily. As an example, for example, if we want to try to study the effects of dexmedetomidine as an adjuvant in the spinal anesthesia, if we make a title that dexmedetomidine, it helps to prolong a dexmedetomidine adjuvant, it prolongs the analgesia in TURP. This could not sound an appropriate title. But if we write the same title, effects of intrathecal dexmedetomidine, first of all, the intrathecal word, it gives what's the route of administration of the drug as an additive. Again, additive conveys that it's not the primary drug that we're using. The primary drug that we're using is the bupivacaine. To low-dose bupivacaine in patients undergoing transurethral resection of prostate. And the last three words, they indicate the nature of the surgery where we are giving this drug. So it may form an appropriate title which could be helpful to the reader. The title contains significant information. Now, if we come to the abstract is the second part that is followed after the title. The abstract, it has to be concise, and this abstract, it helps the readers to decide whether, once they go through the abstract, it helps them to see whether this abstract is relevant to their work or not. Now, most of the abstracts, they are written according to the journal's guidelines, and most of the journals lay down that the abstract, it should not be more than 250 words. And we should always remember that we do not have to add any reference in the abstract. As a reviewer, as an associate editor of some journals, many journals, particularly in the Indian Journal of Anesthesia, we get a number of articles where some of our novice authors, what they do is they put references in the abstract part. And unfortunately, in the first instant, we have to send the article back for the re-revision so as to correct it. Now, once we form the abstract, we should remember that these four main things, they have to be covered. The background and the aims. It should be covered in the abstract. It should be clear as what is the need for the study. And once we form a structured abstract, the second point is the materials and the methods. The materials and the methods, it should again cover as to what is the nature of the design of our study. Is it retrospective? Is it prospective? What are the parameters we are measuring or we are observing? And what is the statistical analysis? We have to give a brief summary in the materials and methods, and we have to give an idea about the results. And we have to shortly communicate what are our salient conclusions that we have obtained from our study. Abstract is followed by the use of mesh words, and usually most of the journals they require that we have to put around 3 to 10 keywords along with the abstract that could reveal the main topic of the study. So once we identify the mesh words, we should keep into consideration that we should take appropriate mesh words from the National Library of Medicine. So at a later stage, again, once any other author who is interested in retrieving our article, who's working on a similar field, he's able to find our articles. So what we do for this? We can follow mainly two strategies. One of the strategies is we can use the mesh on demand, and the other way is we can use the mesh browser. So if we follow the mesh on demand, this is the URL, what we take, and once we paste it into the web browser, this is the web page that opens from the National Library of Medicine. So what we do further, this is one of the articles I have just worked on for demonstrating to our viewers. So what we do is, I have taken the abstract of this article from the Indian Journal of Anesthesia. This article was published in the February issue. And in this article, the authors, what they are trying to do is they are trying to study the analgesic efficacy in the post-operative period of the pulmonary recruitment maneuvers. And they are comparing it with the intraperitoneal hydrocortisone in the laparoscopic gynecological surgeries. So once I take the abstract, put it, copy and paste it into this box, on the side, I get a number of mesh terms. Once I go through them, I am getting the mesh terms in the form of humans, saline solution, hydrocortisone, carbon dioxide, pain management, visual analog score, analgesia, laparoscopy, and demography. So out of all these mesh terms, I select the appropriate mesh terms that are relevant to my study. For example, analgesia could be one of the appropriate mesh terms. Laparoscopic gynecological surgeries or laparoscopy could be the other appropriate mesh term, and hydrocortisone could be the third appropriate mesh term. The other way is to go through the mesh browser. Again, I take this URL, paste it on any web browser, and once I reach this page opens, medical subject headings 2021. I have taken the same article, and from this article, I have taken the one of the leading terms, that is pulmonary recruitment maneuver. And once I paste this pulmonary recruitment maneuver and go for searching of all terms, I'm getting many results, around 1,000 results. So I have just taken, for the sake of clarity, these various results: chemokine, lung disease, again fungal. So if I analyze all these results, pneumonia, pulmonary adenomatosis, on the first page, I am not getting any relevant mesh terms that are relevant to my topic. So I'll continue my search, select the appropriate mesh terms, and incorporate them as the mesh words. So after writing an abstract, how do I confirm that I have formed a proper abstract? If I'm able to answer these 4 questions, why did we start doing this study? What did we do in our study? What did we find? What were our results? And what was our main conclusion? What does it mean? So if I'm able to answer all these 4 questions, probably this may be one of the appropriate abstracts on which I can work around. The second part of any scientific writing is the introduction. So introduction, it should state the main purpose or the rationale as to why we are carrying out this study. More important, again in introduction, is what is the background of the problem. So I have to analyze what is known about the subject and what is not known about the subject. For example, if I want to work on the role of hypothermia as a cerebral protective measure in head injury patients. So if I have a brief summary of the subject, I know hypothermia may not be beneficial as a cerebral protection measures, as a cerebral protective measure. But if I further analyze the subject, it may not be known whether if I take patients who are subjected to mild hypothermia and another group of patients who are subjected to moderate hypothermia, another group of patients who are subjected to severe hypothermia, what could be the outcome in all these 3 groups? Or if I take patients with mild hypothermia who are having a midline shift, and again I take patients with mild hypothermia who are not having a midline shift, how they will behave. So I have to work on this area and mention as to what is not known about the literature in my area of work. And this will again help me to highlight the gaps that are existing in the area in which I am conducting the research. And again, then I have to give a brief introduction. as to what is the relevance of my research problem at the local, national, and international level. Head injury is quite common in Srinagar, quite common in northern India, quite common in India. We are the second most, we have the second most in terms of incidence. We are having the second most common head injuries in our younger population. And again, at the international level, we know that head injury is a global, silent global epidemic. So it is one of the most relevant clinical research questions that needs to be answered. And again, in the introduction, I have to see what are my primary and secondary outcomes that I am studying. In the so how do I work on the primary outcome? Primary outcomes, they are the primary research goals that I will work on, and the secondary outcomes, they are mainly the allied research goals. For example, if I plan to do a study on COVID-19 vaccination and try to see its efficacy, so my primary research goal could be to study two groups and see the infection, the incidence of COVID-19 infection, and compare it among the vaccinated and the unvaccinated patients. While as my secondary research goal could be to see what is the cost effectiveness of this vaccination program. So before I conclude the introduction, my primary and the secondary research goals should be very clear. And whatever important statements are made in the introduction part, they should be backed by pertinent references. Many a times we do get many references, introduction which is filled with too many references. So we should try to avoid too many references into the introduction part. Though introduction, there's no word limit for the introduction, but it would be better if the introduction has only 10 to 15% of the total word count of the paper. Materials and methods, it is the next part that we concentrate after the introduction, and it gives a brief idea about the methodology of the paper that we follow. And this is one of the most important parts that the reviewers and the editors judge before they decide about the acceptability and rejection of the paper. If there are some methodological fallacies, they are uncorrectable and the paper is very likely to be rejected. The other things may be correctable, but if we have some methodological issues, so this is a part where there should be no discrepancies before we proceed on to conduct the study. Once we start writing about the material and methods, we should again make a mention whether we have taken an ethical clearance from the institutional review board, or if we are studying some drug, we have to have a pharmacovigilance clearance. And more important is, have we taken a patient's consent? Then again, have we obtained have we registered our study with the clinical registry of India? That is again to be mentioned. And sometimes some of the authors, novice authors, they try to disclose the identity in this section. The identity of the authors and the institutes has not to be disclosed in this section. Again, in the material and methods, we should decide what were the inclusion and exclusion criteria. They should be clearly defined. And if we are using some scoring sheets or scales, we should use only those scales which are scientifically validated. For example, if we are using pain scores, we should use a scientifically validated score like a visual analog score, or we can go, if we're trying to see the sedation scores, we should go for a proper sedation score, which is scientifically validated. Again, in the material and method, we should work out on the sample details. And sometimes some of our novice authors, what they do once they calculate the sample details, they calculate it on the basis of some study, but the primary objective of our study is different, and the primary objective of the disease, so of the study they quote, that is different. So once we try to calculate the sample size, the primary objective should be, if we are taking a reference study, should be as close as possible to our study. Then we have to have mention about the exposure or the intervention that we are investigating. And similarly, what we have to have clearly mentioned, what are the outcome measures that we are studying in our study. Again, in this section, we should clearly mention that the software, what we are using, and the statistical tests, what parametric, what non-parametric tests we are using for our study, all these things should be mentioned clearly in the material and methods section. And we should not forget to mention what was our study period. When did we start the study? When did we complete the study? What was the place where it was conducted? We need not reveal the place, but we can mention whether it was a primary, secondary, or a tertiary care center, or which part of the country was it performed in northern India, southern India, western India, or the eastern part of India. And more important, have we done a proper randomization? Have we done an allocation concealment, and what was the technique of blinding? All these things need to be mentioned in materials and methods section. The results section, it forms the heart of the manuscript. It should be written in the past tense, and it is the results section that confirms or rejects the hypothesis that we had initially built. So it would be better if we present our results section in the form of flow chart, and we show the recruitment procedure that we have followed. It is ideal if we analyze the data critically, and then we use the various subheadings to present the data. And once we compile the results, at the end of the compilation of the results section, we come to a definitive conclusion. Have we been able to answer the research question that we had raised at the beginning of our study, or it's still incompletely answered? So it gives us an idea that do we need to carry out further studies in this direction that could be helpful in arriving to better conclusions in the field of interest that we are doing the particular study. Coming to the discussion part, many times the articles from novel authors, the novice authors that we get, the discussion is a rewriting of the results. The discussion, the result section should not be repeated in the discussion part. So it would be better if we could start the discussion part by answering the research question in the first paragraph itself, and then it's followed by comparing it with the existing literature that exists in the field. And again, once we compare it with the existing literature, it's always prudent to select the most recent articles. And it's again prudent that we take these articles from high impact journals. Right now, there are a lot of predatory journals, and it's no fun comparing our results from the articles that are published in these predatory journals. So once we are writing the discussion, once we get any contrasting results to our study, we should look for a scientific reason as to why our studies, as why our results are different from these studies, and provide the possible explanations along with the references. At the same time, we should not forget as to what are our strengths, what are the novel features or the strengths of our study, and at the same time, what are the limitations that we could not overcome in our study, and what could be the possible means of improvement in the future as to overcome these limitations. So if we come to the last part of the original study, we have the conclusion part, and the conclusion part, it should summarize the key message that has been already discussed. Here we should not mention anything that has not been previously discussed in the article. No new thing should be introduced in the conclusion part. And similar to the abstract section, we should not put any reference in the conclusion part. And at the same time, we should not attempt to overstate the importance of our findings in our research. And we should try to avoid reaching to erroneous or statistically unsupported conclusions once we formulate the conclusion part of the study. References, once we formulate the references for our article, they should be formulated as per the journal's instructions. We follow mainly the Vancouver style, and use of et al. As per the ICMJE guidelines. We follow the et al. After 6 authors. If there are more than 6 authors, we put the et al. After that. And we should not again forget about the tables and figures. These are one of the most important parts that the reviewers and the editorial board meticulously looks into. And a good table and a figure, they save a lot of unneeded explanations and lengthy discussions in the text. And again, the figures in the text, they show important trends and simplify the detailed data in the textual parts of the scientific paper. Then there are some supplementary parts we need to take care of. Once we submit any article, we have to fill in the title page properly. And there again in the title page, we should put in the title of the manuscript, the type of the manuscript, the type of the study, and then we should not forget to write the names of all authors along with their institutional affiliation and contribution of each author. And again here in this section, we should put in the registered trial number and the running title, and we should put in this copyright form which has to be signed by all authors. So once an editor or a reviewer looks at a manuscript, what are his expectations of a good manuscript? I'll just try to summarize in this slide. If we look at the title, it should be very specific and it should be descriptive. It should cover the main features of the study. The abstract, it should be again specific and it should be within the word limit. Introduction should focus on the background. Research questions should be stated clearly. Literature should not be too old. Many a times we get very old references once we are reviewing the articles. The methods, they should be descriptive and they should be replicable easily at any place. Figures and tables, they should be appropriate. They should be properly labeled. Legends should not be missing. Citations should be properly described. And once we look into the discussion should focus mainly on the research question. There should be an appropriate comparison with the studies that have been done earlier in the field. We should always mention the limitations of our study and what are the what's the scope for the future research. And all the writing of our scientific paper, it should be logical. It should be arranged in a logical way. An editor's perspective, once we have a look on the paper, mainly incorrect style is not liked by any editor or reviewer of any journal. The chances of acceptance get diminished considerably. And once we submit any paper, we should recheck the paper and correct spelling errors, punctuation mistakes, grammatical and syntax errors. And again, we should take care of the figures and tables, and we should recheck the data, the numericals, for their consistency and accuracy. Once we submit the article, nowadays there's a lot of pressure among most of our colleagues for publications. And if we want to publish quickly, it may lead to rejection. Most of the reviewers and editors across the world, they do work on an honorary basis. So they do expect good conduct from the author. So once any author submits any manuscript for a journal, we expect some patience for the review process and the decision. And once we submit any article to a journal and it comes back for a revision, the clarification should be given for by the authors on a point-to-point basis. We should not casually attend to these comments. If we go to the systematic reviews and meta-analysis, they just follow a more structured protocol. If we go into the systematic review, they follow the PRISMA checklist. It consists of around 27 checklist items, which are based on the appropriateness of the title, the abstract, where we have to have a structured summary, then the introduction, what is the primary and the secondary objective of the study, the methods, where we follow the, has the protocol been registered, the eligibility criteria, the search process that we have formed, the study selections, the data collection process. And once we again go into the results, we have certain subsections as how we have done the study selection, the study characteristics, what is the bias within the studies, what are the results of the individual studies. And again, in the discussion, we have a look on the summary of the evidence, then the limitations and conclusions, and lastly, what we have to see what is the funding, was any funding source utilized for conducting these studies. One of the commonly written publications, scientific communications, is the case reports. And they help us to publish the new unusual conditions or etiologies in the literature. So once we publish the case reports, it's better to, before we publish the case reports, it's better to follow the care checklist. So here again, we see the appropriateness of the title once we publish the case report. And again, have we taken an appropriate keywords? And then once we form the abstract of the case report, in the introduction, we should mention as what are the unique features of the case report. What new does this case report add to the scientific literature? And again, once we make a conclusion in the case report, what are the takeaway lessons from our case report that we have published or we are attempting to publish? Again, into the introduction part, we have to make it very clear that's why this case is unique. Then once we provide the patient information, we should give a brief idea of the primary concerns or the symptoms of the patient. And in the clinical findings, we should not forget what are the significant findings in the physical examination. And then we should give a brief timeline of the patient's history to the point where we have given him an appropriate care in our setting. And then what are the diagnostic assessment techniques we have used to arrive at a particular diagnosis, and then what is the therapeutic intervention we have done. And These are some of the points. Again, we should not forget once we formulate a case report as to what was the outcome that was seen once we have reported this unusual case report. And then again, in the discussion part, we should mention, are there any similar case reports in the literature? What is the rarity of the condition? How is our case report different from these previously reported cases, and what are the takeaway lessons? And if possible, we can even put a patient perspective on the treatment that the patient has received. And last of all, we should not forget to take an informed consent from the patient. Thank you. Thanks a lot.
So, Dr. Julfikar, can you stop sharing your screen, sir?
Yeah.
Over to Dr. Kirti, ma'am, for the next speaker. Can I request Dr. Divetia, sir, to kindly introduce the next speaker, please, sir?
Okay, so the next speaker is Dr. Kapil Dev Soni. He's an additional professor at the Jawaharlal Nehru Apex Trauma Center of AIIMS, New Delhi. And he is a fellow of designing clinical studies and evidence-based medicine at AIIMS and a visiting faculty at the University of Michigan, Ann Arbor in the USA, and affiliate of the ANZICS Research Committee in Australia. He's of course he's an expert in trauma research and trauma critical care, and he's a member of the group for standard trauma guidelines for India. He's a co-investigator and a member of steering committee of many projects, and he's published multiple over 100 papers in national and international journals. And he will be speaking to us on a very important topic of critical appraisal of a published paper regarding therapy. So how do we read papers that have already been published? So Dr. Kapil Dev Soni, please start.
Thank you, sir. Good evening, all. I'm Dr. Kapil Soni, and I would be speaking on critical appraisal of published paper. And specifically, I would be speaking on critical appraisal of a therapy paper. Now, whatever till you have heard from our previous speakers, that's what about the writing scientific study or designing a scientific study. But this is important for the consumers of the science, that how do you critically appraise a published paper? And with the today's day and today's era, you will see a lot of publications coming in the PubMed, and a lot of the publications, most of the journals are publishing. But remember, not everything which gets published is credible. So your job as a final consumer of the science is to know whether the published findings are credible or not. And how do you do that? Well, I start with a case scenario. Suppose you are a 55-year-old male who had a case of a chest injury, admitted to ICU, and develops a acute respiratory distress syndrome and requires a mechanical ventilation. And in spite of supportive therapies, he deteriorates further. A registrar on rounds asks you, or as a consultant, that should we prone the patient in addition to other supportive therapies? So this is completely a clinical question. And as a rounding consultant, you would say that, okay, just go and see what are the what is the published evidence about this prone position. Well, when you begin thinking about answer to a clinical question, you go from 4 stages. One is you ask a particular question, you acquire or you search the published evidence, you appraise the evidence, and you act on the information which is available in the evidence. So what is the question here? The question here is, among adult ICU patients who develop severe ARDS, does the application of prone positioning reduces mortality? So this is a classical question which is pertain to therapy of the ARDS, right? So the registrar goes back into the literature and sees there was a paper which was published in New England Journal of Medicine that was about the prone positioning in severe acute respiratory distress syndrome. And he reads simply the conclusion, which suggests that, okay, in patients with severe ARDS, early application of prone positioning is beneficial. It reduces 28-day and 90-day mortality. He comes back to the consultant and says that, okay, he has found the evidence and we should prone. Now the consultant actually asked him, is the findings credible? Can we believe what is published in the literature? So he says that, okay, to the resident, that go back and critically appraise the paper. A critical appraisal means you have to assess the paper in 4 domains. One is whether is it relevant for your clinical practice? Is the study which has been published is valid or not? Are the results adequate and appropriately described? And whether the results are applicable for your patient management. So he looks at the abstract, which suggests that, okay, yes, it's quite similar to the patient or to the clinical question he's trying to ask. So how do you decide a paper is relevant for your clinical practice? You look at the 4 things. One is population, intervention, comparison, and outcomes. If the paper reports a similar population or a similar intervention which you are unsure about, and a comparison which is a standard practice of yours, and the outcomes which you are interested in, so that paper becomes relevant for a clinical practice. So if you go back to the manuscript, you will find that where would you find this information? You will find information in the method section that would describe the patient, the intervention, the comparator, and the outcome. The patient population in the published paper was around 474 adults with severe ARDS from 26 ICUs in France. Intervention was a prone positioning for at least 6 consecutive hours, and the comparator was a supine group, so which is quite relevant in the ICU population. He also looks at, well, what was the primary outcomes. The primary outcomes which were reported in the paper was the mortality at day 28, and then there were numbers of secondary outcomes. The mortality at day 90, the rate of successful extubations, the length of ICU stay, the use of NIVs, the complications, tracheostomy rates, number of days free from auger dysfunction. So they were the secondary outcomes, right? So going back to how do you critically appraise this paper now? First of all, we must be familiar with the basic design of a therapy paper, which is the randomized control trial. It's a prospective design. There would be intervention, there would be treatment, there would be placebo controlled, there would be randomizations, there would be blinding and follow-up of participants. But to critically appraise, the validity is the most important aspect which you would be looking at. How was the study performed? Can I trust the results of the study? And after trusting the results, can I apply the same intervention to my own patient, which I see day in, day out? So a validity, when we talk about validity, there are 2 types of validity. One is the external validity, other one is internal validity. And which one is most important for you? For a practitioner, both validities are important, but the internal validity has got more preference. If the study is not internally valid, then there is no question of external validity. So when you are appraising a therapy paper, you look at the internal validity. In internal validity, you look at 3 stages: whether the study started well, whether it was run well, and whether it finished well. Start well means you look at the control group, whether the study had a control group. Then you look at the concealed randomization. Then you look at comparability of group at the beginning of the study, whether the group were prognostically balanced or they were imbalanced groups at the start of the study. Then you look at how the study went on. How did it went? You look at the co-interventions which were done in the control group and the co-interventions which were done in the intervention group. You look at the contamination, how many patients they actually crossed each arm from intervention to the control and from control to the intervention. You look at the compliance of the intervention. How much was the compliance of the intervention? And finally, you look at how did the study finished, whether there was a complete follow-up, whether there was a correct outcome measurements and assessment, and whatever analysis which has been reported in the paper, is it credible or not? So these are the what I say is 9C when you are looking for internal validity. Well, Dr. Rakesh has already pointed out that a control group is important. Why a control group is important in a therapy paper? If there's no control group, then there are alternative explanations for the beneficial effect. It could be simply a natural history or a Hawthorne effect or a placebo effect or effect of confounder, co-intervention, or a bias. And that's why the control group is important. How this control group is created, that's also important. So that means there should be a randomization and concealed allocation. Now, what does a randomization do in a therapy paper is that it actually balances the known as well as importantly the unknown prognostic factors. Now, with our knowledge, what we know about the disease, we know only about 50% of prognostic factors, and 50% of most of the diseases or prognostic factors are actually unknown. That's why a proper randomization is important. Randomization actually distributes the known as well as unknown prognostic factors almost similar in both the groups. It creates a prognostically balanced groups at the start of the study. So if you're looking at the paper, you would look at whether it was a randomized or not. Secondly, was it a concealed allocation? Now, why this important concealed allocation? If the person who is enrolling participant and he knows where the patient is going to go in whether it's going to go in control arm or it's going to go in intervention arm, he can actually distort the relationship. He can introduce a bias. And what we have seen that the trials which do not report allocation concealment, they have larger estimates of treatment effects, which are actually untrue, which are not true. So concealed allocation is important. Going back to the paper, if you look at the paper, there was a randomization. There were around 1,434 patients were screened, and out of that, only 576 were eligible. And few of them, they were excluded. 474 underwent randomizations, and 234 and 240 in a supine and a prone group were randomized. Second part in a critical appraisal is whether of a randomized control trial is whether during the study period were they comparable. So you look at the baseline variables, prognostic balance, and whatever differences are there at the beginning, you attribute that differences to the chance. That's why we say that in a properly randomized control trials, most table 1 will not have a p-values because we believe that the randomization has worked. Back to the paper, if you look at the table 1, which is basically a comparison of two groups, supine and a prone, and there's no p-value which has been described. Now, this is a standard practice in most of the randomized control trial that the differences between the two groups are not reported at in the table 1. And this is because we believe that randomization has worked and it has created a prognostically balanced group. So these are the initial parameters of the two groups, supine group and the prone group. The variables, they were quite similarly matched on both the groups, both in the demographics as well as the ventilator settings as well. Now, during the study, we look at what were the co-interventions and whether the co-interventions were similar in both the groups or there was unequal care. Then you look at the contamination, how many crossed each other. Now, what happens is that if because of contamination or crossover of participants from intervention arm to the control arm, there would be underestimation of effect, and this would bias the results towards the null value, right? Now, finally, once the study gets completed, you look at the compliance, the efficacy of the intervention, the tolerance, and the safety. Complete follow-up. Now, most randomized control trial would attempt to have a complete follow-up of all the participants, but in real world, this is not achievable, and certain studies would have loss to follow-up. So what to do when you have a loss to follow-up? How do you decide whether the results which has been reported are actually close to the truth or not? So one way to do that is that you do a sensitivity analysis, you assume a worst case scenario, you assume that whomsoever had lost to follow-up, they had a bad outcome. And then you reanalyzed the finding. And if the finding remain within the same direction and may not be the same magnitude, but the same direction, then you trust the results. Second one is what about the outcome measurement, whether the outcomes were subjective or objective? In this study particular, the outcome was objective, a 28-day mortality, which is a very hard outcome. Mortality cannot be ambiguous, right? And when you have to look at whether there was a credible analysis or not. And how do you decide whether there was a credible analysis in the study? You need to look at the ITT principle, which we call as intention to treat principle. Right. Intention to treat principle is that you analyze, or the study actually reports either the per protocol analysis or intention to treat analysis. That's based on if the patient has been randomized in intervention arm or a control arm, and he should continue to be analyzed in the same arm, even if he has crossed over to other arm. So intention to treat means analyze as randomized. Now, this is because if you go for a per protocol or you allow contamination and you analyze as based on per protocol, that would actually distort the effect of randomization. That would introduce a bias towards null. So when we say intention to treat, we assume you analyze as randomized, irrespective whether after randomization, the person or subject has received intervention or not. So back to the paper, if you look at the flow diagram, the consort diagram, 474 underwent randomizations, 234 were in the supine group, 240 were in the prone group, and then there were certain exclusions, and 466 were included in intention to treat analysis. So this is what you would be looking at if you want to trust the finding. What are the threats to internal validity? So there are broadly three types of threat to internal validity. One is a chance variation, a random error. And that means if you see a result which is a positive, it might simply be a chance finding. A chance finding means this could just because of a chance you have seen a beneficial effect of intervention. It could be because of a confounding, or it could be because of bias. Confounding and bias are systematic errors. Systematic errors, what I mean is that they are introduced during the conduct of the study, whereas chance variation is just a pure chance, right? So how do you decide whether the results which you have seen in the study are because of a random error or they are because of a systematic error? For chance or random error, what you need to look at is the confidence interval, which provides a sense of precision, and the test of significance, the p-values. Statistical inference is used to understand whether observed results are due to chance or not. Just by looking at confidence interval or p-values, you would be able to identify how much chance is playing the role. Now, I must say that when you see such results, you need to look at the statistics which has been applied during the presentation or in the manuscript. And if you see there is a role of multiple testing which has not been accounted for. So the observed result might still be because of a false positive findings. So what is confidence interval? It's just how precise are the results. This is important because if sometimes you may not see the desired result, that might be because your sample size is too low or do not have a power to show the result. And this is example study of 100 patients per group and the study of 1,000 patients per group. Obviously, the study of 1,000 patients per group is more precise and it can actually is able to show a true beneficial effect of intervention, whereas a study of just 100 patients per group, because of low power and low precision, is unable to show the true effect. So when you are appraising a paper, you need to look at how precise are the results, what is the confidence interval, is it enough powered to show the result or not. Sometimes studies are underpowered and they do not show the benefit effect of an intervention. So that need to be taken care of when you look at confidence interval. A systematic error maybe because of a confounding, and confounding is, we all know that this is because of other factors which distorts the relationship between the exposure and the outcome. So you need to look at what are the confounders in the published paper. And finally, you need to look at bias. What is a bias? A bias is basically nothing, but it's simply a deviation of a truth which is beyond chance error. There can be deviation from the truth. Truth is one, but then the results might deviate from the truth. That may be because of chance error, or they may be because of a bias as well. Systematic errors, which has been introduced in the study by the investigators in sampling, collecting, or interpreting the data. So there are broadly two types of biases. though if you can go into the depth, you may find almost 64 types of biases, but in broadly there are two types. One is a selection bias. Selection means the distortions that arise from procedures used to select subjects, or factors that influence study participation, or the factors that influence participant attrition. So that's a selection. How the subjects have been selected for the study, how they are maintained in the study, and after follow-up, how they are lost to follow-up. So that causes a selection bias. Second bias is basically a misclassification bias. That means classification of exposure and outcome. Drop-ins and dropouts would bias the association towards the null, whereas classification of outcome can be happen because of subjective outcomes. But if they are objective outcomes, they are more credible. So internal validity, you can look at whether the studies have actually done all these safeguards to prevent or threat to internal validity. So one is there should be randomized, intention to treat, there should be excellent follow-up, there should be blinding, adherence, and outcome should be objective. So this is how one can take care of the threat to internal validity. Blinding is important because if the groups are not blinded, most trials would throw up large estimates of the effect. Whereas properly blinded trials, the effect estimates are quite narrow. They may not be that large. Blinding, who can be blinded? A patient can be blinded, the clinicians can be blinded, the data collectors can be blinded, adjudicators can be blinded, and data analysts can also be blinded. So these days we have stopped using terms of single blind, double blind, triple blind. What we say is that we ask specifically or look into the paper specifically who were blinded. And if they were not blinded, how much bias it can introduce in the paper or in the study. Results, there are a number of ways to express the results. One is the risk difference, number needed to treat, risk ratios, odds ratios, margin of error, or time to event. So there are a number of ways to present the results. So back to the paper, of the prone positioning, randomization was computer generated and stratified according to ICU. You can have a great methods here. Outcomes were mortality, and then there were subsequent secondary outcomes. Now, importantly, when you look at the outcomes for believing whether the results are credible or not, you want to look at whether the outcomes were adjusted for important confounders. So here the authors have adjusted for SOFA score. A SOFA score is a severity score in the intensive care patients. And if you look at those two scores, not adjusted and adjusted, you see how much variation is there. If there's not much of variation, you still believe that, okay, even after adjusting for SOFA score, the results or the magnitude of effect remains almost consistent, similar. If there is large variation, then you would have doubt over the published results. Right? So similarly, you have to see whether the paper reports adjusted analysis as well or not. The magnitude of effect in the reported paper is a mortality difference of 16.8% of the prone positioning, which brings a number needed to treat around 6 and risk ratios of 0.48, which is a huge effect of the prone positioning. Paper also report about the adverse events. In a therapy paper, when you are appraising it, you should also look at what are the side effects which have been reported and what are the adverse events. A therapy paper is incomplete if it does not report adverse events. Applicability assessment. So once you are certain or you have confidence over the estimates of the reported paper of the findings, you want to know whether the results are applicable to your patient or not. And you have to assess whether the patient is similar to what has been reported in the paper. That means the PICO and whether the benefit outweighs the harms or not. If the harms are more than the benefit, obviously the results are not applicable. So you have to again balance the benefits and harms of the published intervention effects. So what conclusions have been drawn in this paper? In this paper, the authors say that patients with severe ARDS, early application of prolonged prone positioning sessions significantly decreased 28-day and 90-day mortality. So when you're appraising it, you would suggest, okay, whether their conclusions is justified by methods and the findings. And you would say, yes, the methods are quite robust and findings can be trusted. But can you trust such amount of magnitude of effect of the intervention? Well, if you look at the paper back, the paper actually had a lot of screening, and out of that 1,500, only around 500 were included. So why 1,000 patients were not included after screening? So that may introduce a selection bias. Then if you look at the first table where the prognostic balanced or start of the therapy, the groups were compared. You look at found the SOFI score, which was 10.4 in the supine group and 9.6 in the prone group. It means the severity was quite lesser, but we don't know whether it was significant or not. But if you look at the vasopressors, there was 83% vasopressors in the supine group and 72.6% in the prone group. Definitely the prone group was slightly better prognostically at the start of the trial. And then there was a higher use of neuromuscular blockade. Most importantly, this study, all the centers had more than 5 years of proning experience. And when you want to apply your finding of this study to your center, you need to look at whether your center has some experience of proning patients or not. So then the question of applicability will occur. So if you look at the first table where they have report, you find that there is a certain discrepancy and it may bias or may enlarge the true magnitude of the effect of prone positioning. 16.8, which is quite big. The true, maybe it might not be 16.8, it might be somewhat lesser than that. But yes, these biases may expand the magnitude of effect. So coming back to the paper, coming back to the question which the resident has asked to his consultant, should we prone the patient in addition to other supportive therapies? The answer is yes. Patient is similar. All important outcomes are considered, and the benefits are more than the harms and cost. So there are a lot of ready to do therapy critical appraisal cheat sheets. You can, if somebody wants to critically appraise a paper, they can go through that cheat sheets and see whether the studies are appropriately conducted and whether there is a internally valid or not. And once they are internally valid, only then the confidence in the estimates can be maintained. So for critical appraisal, relevancy assessment is important, validity assessment is important, results assessment is important, and applicability assessment is important. Thank you. Thank you for patient listening.
Respected Chairperson, I think we have finished all the 4 sessions, so we have some time. So may I request Professor Mohit to initiate the questions session, and any questions from the participants they can also ask by putting up in the chat as well. Over to you, sir.
Thank you, Dr. Rakesh. It's a pleasure and honor to be there, to be a part of this program. It's an excellent program, and I must thank the dignitaries of ICA for giving me this honor. We have certain questions in the chat box. Some questions. First question is, do we need to? Can we perform an RCT in a nursing home? I think can I ask request Dr. Rakhi Goel to if she can put some light in this this question.
Yes, please. Of course, we can do. I replied to the person who asked separately that there are independent ethics committee there in every sector of the country. So they can check online which is convenient to them, and they can apply their protocol to the ethics committee. And if they approve, then they can get their trial registered and go ahead with the trial. So there is basically the whole concept is that nobody is should be everybody should have an access to do a research study. That's the whole premise. So yes, the answer is yes.
Yeah, yeah, absolutely on the dot. It should be like, yes, any study, whatever you're doing, it has to be, I mean, it has to be registered. At the same time, ethical clearance needs to be taken before, because you are trying to perform any sort of trial on a subject, on a patient. So naturally, the consent along with the ethical clearance needs, it's mandatory. And I think every small institution, whatever it may be, of whatever category degree it is, they must be having an ethical committee of their own. The second question is, which is really, it's something which is very common, and I think most of the institutions they have. What is, do we need to have a CTRI? I mean, registered for a thesis, MD thesis. I mean, I think I think if Dr. Rakhi Goel can again answer this question.
Yes, certainly
Yeah,
yes.
yeah.
They all must get it registered so that tomorrow when you present somewhere, you publish. That's the aim. Ultimately, you want to publish in a good journal so that people can read your work. So you must register prospectively before the recruitment of your first case. That is important. People think that they'll register later. So then they lose out. CTRI does not register retrospectively. So they should register prospectively before starting testing.
I think we don't have much question on this. Then I have a query. I think, of course, it's a query which can which everyone will have. These CTRI has been very strict and stringent after, I mean, right for the last 2 to 3 years, especially in journals. They need the CTRI, especially after 2018. They made it mandatory. If anyone who wants to publish his article or her article, which has not been registered, so what do they do then in that scenario? Supposingly he's done a study which was in 2017, 2016. He wants to get it published now. So how does he get it published? Because nowadays journals are, yeah.
I think
So
we have
the editor of the journal can give a waiver, as I understand, but CTRI just does not register retrospectively anymore.
we can directly listen from the editor-in-chief. We have Dr. Anju
Yes.
Grewal and Dr. Divetya. I think
Yes.
how they will handle the situation when somebody says they don't have CTRI registration.
I think this is a very common question, common finding, because I've been also dealing such questions in the journal as well, because they also come up with these problems to us, and at times I also find how to handle these questions. I think Dr. Divetya will be very, confident on these questions.
So Dr. Moid,
Yeah.
if I can answer that, so you have to find a journal which does not insist on trial registration. Okay, and as of now, Indian Journal of Anesthesia still takes unregistered articles. I don't know what is JOACP stand now, but very few journals now take unregistered trials. Okay, so
Yeah.
you have to literally search for a journal, or you may write to the editor and say, "Will you consider?" But invariably, as soon as you don't have a registration there, they will send it back to you. So you need to look for your journal. The other thing about CTRI registration is, very often when you're doing a thesis or even if you're doing a proper study, you are a little careless about CTRI registration. You just say, चलो कुछ को डाल देते हैं, भर देते हैं, फिर हो जाएगा, उसके बाद study शुरू करते हैं. If you ever send this article, this paper afterwards to a good journal, they will compare what you have written in your primary aims, primary objectives in your article with what you registered in CTRI. The method that you described in your paper with what you were in CTRI. So it must be matching. You can't just, just jaldi
Yeah, that is true. It's very transparent. You just cannot fool people.
Matching, and I have known of good journals which have rejected articles saying that this is not what they've registered in their, on the trial registry.
Absolutely. So that's exactly why we are having these kind of CMEs, where we are trying to educate our young postgraduates to do it the right way. Repeatedly, we are telling them that this is important. Just an hour or two it takes to register. Please register.
Yeah, as at JOCP, we had two editorials touching on the same topic. And we are, of course, very insistent on getting a CTRI. We have been trying to follow that as strictly as possible for the last 2 years. But if there is an answer to Dr. Moed's question, is if we would actually look at the content and the validity and the merit of the article, and then of course maybe a waiver might be given, but usually we will definitely need a CTR. It's better to register so that transparency of your research is maintained more than the publication. I believe it is the research for which CTRI was basically introduced.
And as for the current guidelines, article on COVID has to be registered with CTRI, even if it's observational, even if it's retrospective.
Yes.
If it's COVID related article, it has to be registered.
Absolutely. Dr. Rakesh, if I'm allowed, just a query from one of my postgraduates to you.
Absolutely, ma'am. It's wonderful to listen to you, ma'am.
Sometimes ethics approval committees would insist on change of a study design for them. Though I believe and I understand that ethics approval. I think I got muted. Ethics approval committees are primarily, designed to look at the ethical aspects so that no harm is coming to the participants. But there have been times where a change in the design comes in, much to the, disillusionment of the person who is actually giving it. So, how do you would propose them to, go ahead and solve that issue? A little bit of an ethical dilemma.
I know it's a difficult question and practical also at times, because there are 2-3 options. The best thing is, which I've even learned from you only, the best step for a successful publication is to start well. And start well means when you're making a protocol. So it's the best time when you're making a protocol, maybe when a postgraduate student is doing it, it's always good to share the experience of seniors that they are going to use this technique. Is it going to be having some constraints in that particular setup, because they know everything, they have been working in that setup. So that's why once you are aware of it, so when we talk about evidence-based medicine, it's just not the published literature, but it's the local experience also for safe conduct of that particular aspect. So involve the seniors, take their opinion before you submit.
Sir, please, आप अपने दोबारा retype करिए क्योंकि मैं disconnect.
Dr. Anju.
Yeah. Yes, sir.
So we've had these discussions. I've been on both sides as a researcher as well as a secretary of our ethics committee.
Okay, sir.
So the argument of the ethics committee is that it is not probably ethical or not correct to do a poorly designed study, right? And so we will look into some aspects of study design, especially the views of lay members, especially the views of social scientists and people. So, and at the end of the day, you need ethics approval. You might as well do what they're saying, unless it's something grossly wrong or they've completely misunderstood the protocol or something like that. But otherwise, it's best to just give in. Okay.
I agreed with you. Fully agree with you. Indeed, why I've asked you this question is this came from one of my postgraduates who was actually told to change the study design. And then what exactly Dr. Rakesh said is, and finally we gave into that ethics committee, I believe that group gave into that committee. So we
So ma'am, if I may add, we have that scientific committee meeting, which is before the ethics meeting. So the study design part is dealt with in detail during the scientific meeting, because the ethics guys won't really know, especially like our common thesis topics, they don't know what desflurane is, what sevoflurane is, what bupivacaine is. So that scientific committee deals with all this matter and any change in or refining of the protocol, I would say. Not a complete change, a refining, which would make more sense. So that's cleared earlier at a earlier stage, and then it's put up to the ethical committee.
And also to all the students, thank you so much, Raki. You're absolutely correct. And also to all the students, I think the last talk of Dr. Soni was perhaps is something that you need to do a true literature appraisal and critical appraisal of your paper before you select your topics and select your and of course, formulate a thesis, a research hypothesis, and then go on to a research design. So thank you so much for such useful inputs.
Some of the students, I think delegates, they want to know what are the journals, which journals are being indexed by the MCI and NMC. I think I would request them to go to the MCI website, and NMC probably it's all mentioned there, because I mean they're having some queries regarding the society journal, which has been a few years back been incorporated in the index by the MCI, recognized by the MCI. So probably they must be having some query on that. So I would request them to go to the MCI website or NMC website, and they'll find out all those publications, I mean journals which are being recognized by MCI. So that is, I think we can tell them to go for that. Anyone would like to suggest, if you know those journals, because there are 5-6 journals which are being recognized by the MCI or NMC.
I believe they haven't mentioned the names of the journals. I haven't gone to the NMC website as of late, but they do mention what indexing would they prefer. So if so we can have a look at the journal main page where they write about us and indexing information, where you can have a look at the information that the journal gives, and is the journal indexed with the indexing agency that NMC accepts.
Yeah, it's actually the indexing agency which they are talking about, probably, but not the journal, because journal as such, we will not be able to know which indexing they are having. So it's the indexing agencies which are being which needs to be known by these people.
Yeah, so I think they have kind of categorized PubMed, Scopus, so Medline, and these are the kind of, they have taken off other indexing particularly.
Hello. Yeah.
Okay.
I think Dr. Saxena wants to ask question. Yeah, Dr. Saxena, you can go ahead with it.
Right. You know, now I'm unmuted. I think जैसे किसी ने गला बंद कर दिया was good evening.
Yeah,
good evening. Anyway, thanks to Dr. Jashi Sood, you know, I really had to call her. Thank you so much. My heartiest congratulations to all learned speakers. You know, really wonderful presentation. The only thing which I was trying to see was something on systematic review and meta-analysis. Obviously, for paucity of time, I know it is not possible always. So I would just like to add a word on that, because Dr. Rakhi was talking about CTRI, and I would just like to add on CTRI that, you know, recently one of my colleagues had a problem because when she sent the paper for publication, it was something related to, you know, the tab block and, you know, those incisional hernia, but later on she had changed it to inguinal hernia, herniotomies, and, you know, obviously the paper was not accepted. So then she talked to Dr. Jyotsna, is a person at CTRI, Dr. Jyotsna. And then she says, you know, that you can always call me, you know, or you can send a mail, and accordingly you can, in between also, you can change the category of patients. So that is one thing. Anyone is most welcome to contact Dr. Jyotsna at CTRI. That is one. And the second one, which I would like to say, because Dr. Rakhi probably could not talk about, that how to register your systematic review and meta-analysis. Because we have been doing a lot of these meta-analysis, and that's why I thought I'll just say a word about that. It is something like Prospero. You know, Prospero is the site where it's an international prospective register of systematic reviews, where we have been registering, and they definitely take into account the publication bias, using those plotting of the visual inspection of the funnel plot. That's one. And they also apply those Agar and Beck tests for testing the publication bias. So, you know, these registration at Prospero for systematic review and meta-analysis is very important. And, you know, their condition is that the meta-analysis will be analyzed only when you have a number of studies is 10 or more. That's very important. And they have their own method of, you know, that Stata version 12, and, you know, the different R software and all that. So that's one thing which I thought I'll add regarding the systematic review and meta-analysis, because as Dr. Rakesh was saying, it's the ultimate thing. Yes, no doubt, you know, on the top of the prism is there, and yes, that's where, you know, we really follow those Prisma P guidelines. And that's very important, because Prisma P guidelines, on that basis only you can follow that. And you also have to assess the risk of bias using the Pedro scale, which has got 11 criteria. And so that's very important. And the other thing which they have in prospective reviews and meta-analysis is the testing of heterogeneity, which is very important. And that is where they have graded and classified it as, a low level or a moderate level or a high level of heterogeneity. And I think something like anywhere from 0 to 25%, it's a low level of heterogeneity in meta-analysis, and 25 to 50% is taken as moderate level of heterogeneity. And anywhere from 50 to 75% is taken as a very high level of heterogeneity. So those are the things where ultimately then they draw the forest plots and all that. They are generated, to assess the contribution of each study which has been included in the systematic review and meta-analysis. So that's something which I thought I must add. That's something which obviously it is not possible every time. And another thing which Dr. Rakesh was talking about, the cross-sectional studies, I thought recently, they have come up with cross-sequential studies also, because that in the designing, the cross-sequential studies, they combine both cross-section studies as well as the longitudinal study. When you want to compare two or more than two groups of different age groups, over a period of time. So it's something like two or more than two groups of different age groups over a period of time. Then cross-sequential studies, that's the need of the hour in comparison to cross-sectional studies, So that's one thing the only thing which my last one would be on, because in pain, it is so difficult many times to conduct your studies and randomized controlled trials is, of course, Dr. Soni talked about, and Dr. Rakesh also on the placebo. The only thing is the placebo controlled trials obviously are needed when you really want to see and assess the efficacy of any intervention, whether it's a drug or it's an actual intervention, the intervention which is using a needle and all that. So the only thing which extreme care must be taken is to avoid the abuse of that particular option, where the person who is actually receiving the placebo, should not be subjected to any kind of serious or irreversible harm, which is not good, So that's something which we must take into account, that when we are undertaking we have a control group where we have a placebo and we know the placebo effect, but any irreversible harm should not be thrown to the person who is actually involved in the placebo group. So these are the things which I thought. And the other one is on rescue analgesia. Many times you are utilizing a rescue analgesia, but the question is, especially in cancer trials, in cancer pain, and there you find that the patient is already on so many antidepressants or anticonvulsants or anti-anxiety drugs and all that, and so many other things are there which you can't take them off when you are actually doing the trial on cancer pain. So then there may be a lot of confounding factors which are there. So that's one. And rescue analgesia, certain drugs you are using, but then you find that there is both nociceptive and neuropathic component or mixed, and I'm using, say, only diclofenac as one of the rescue analgesia, or maybe, any other NSAID, and, that has taken care of only the nociceptive element in the rescue analgesia, but not for neuropathic, or not for combination of the miscellaneous one. So that's another, problem when you're actually doing a trial on, pain and using your rescue analgesia, the results may not actually be. In fact, there are few statisticians who are actually going to the extent that, that we always take P less than 0.05, and they rather want P less than 0.005. I think everyone knows that, So there will be something which is marginal to 0.05, and then we call it as significant. So those are limitations which are, I believe so, they are very important and must be taken into consideration. So thank you so much. Thank you, Dr. Jayashree and Dr. Saeed, and of course, all the learned speakers. Thank you so much.
Thank you so much for the valuable inputs. I think with paucity of time, we will just say and wind up the program. My sincere thanks to the faculty speakers, Dr. Rakhi Goyal, Dr. Julfikar Ali, and Dr. Kapil Dev Soni. Thank you so much for informative session. And thank you to our eminent chairpersons also, Dr. Anju Grewal, Dr. Jayashree Sood, Dr. J.V. Divetia, Dr. Kirtina Saxena ma'am, and Dr. Saeed Mohit Ahmed for taking the interaction and giving us so many important points and nitty-gritties from your experience also. Thank you so much. I will request Dr. Radhakrishnan, Dr. Jayashree Sood ma'am, and Dr. Baljeet Singh to give us the concluding remarks for the today's webinar on essentials of research and publication. Over to you, ma'am, answers.
Dr. Baljeet, are you there?
Well, I'm certainly there.
Yes, please go ahead.
I thought Dr. Radhakrishnan will be doing it, but anyway, on behalf of Indian College of Anesthesiologists, I think this was, I must first of all compliment and congratulate the speakers for the wonderful talks. The kind of clarity that they have provided on different aspects of scientific research, starting right from Dr. Rakesh, where he clarified all those terms: what is a cross-section study, what is a longitudinal study, what is a prospective study, what is a retrospective study, and all sort of things. I think it must have clarified, the doubts in the minds of the young researchers that we have, particularly the postgraduates or who have just, crossed the line and come to the faculty level at the start of their career. So thereafter, Dr. Rakhi Goel had another wonderful presentation. Then Dr. Zulfikar Ali, and of course the last one was quite a had a lot of information, which at least I must say that that was new to me, and I think I'm going back with a lot of new information, which certainly will be useful to me for teaching, training of the postgraduate students. Well, thank you so much, all the wonderful speakers and chairpersons who conducted the whole program. And then of course the participants who asked questions. Dr. Ashok Saxena had a lot of additions which he thought that were missed, and I think very, very important inputs. And on behalf of Indian College of Anesthesiologists, I thank everyone and congratulate everyone for such a wonderful webinar. I think it's time to call a day, and Dr. Radhakrishnan and Dr. Jayashree Sood, I think on behalf of both of them, I thank you all for being there for this webinar, for this wonderful webinar. Thank you so much, and good night.
Thank you very much, and good night.
Thank you.
Thank you so much. Thank you so much.
Thanch you. Thanks a lot.
Thank you so much.
Thank you.
Thank you,
ma'am.
everyone.
Thank you. Thank you. Bye-bye.
Thank you.
Bye.
Thank you, everybody.
Thank you. Bye, Anju. Bye-bye.
Thank you, ma'am. Bye.